Hi, my name is Samantha Oviedo, and I am a sophomore in the STEAM program here at UTSA. I am a part of Dr. Lindsay McPherson's lab, and the title of my poster is Investigating the Innervation of Taste-Receptor-Like Chemosensors in the Gut by Vagal Sensory Neurons. Have you ever wondered how your brain is able to control digestion? Well, it all begins with your tongue. There are taste receptors on your tongue that transmit signals via vagal sensory neurons to inform the brain of chemical stimuli. In mammals, stimuli can be broken down into 5 primary taste modalities, which include sweet, sour, bitter, salty, and umami. And in recognizing any of these modalities, it's able to discern between nutrients and toxins before they're actually digested. Similarly, sensory like cells line the epithelium of the gut and are equipped with chemoreceptors. These chemoreceptors relay sensory information to the brain via local enteric neurons and are able to control digestion. Some of these cells actually display a similar taste nutrient signaling mechanism seen in the tongue and innervate vagal neurons to elicit post ingestive signals. A variety of vagal neuron types have been identified. Some are in nutrient sensing, some in stomach distension, and some that even induce a swallow or cough. Here, specifically in my project, we are focused on vagal sensory neurons expressing calbindin-1. Calbindin is a calcium binding protein, and this protein has been shown to be expressed in a small subset of neurons innervating the larynx and the trachea. These neurons innervate the epithelial layer where taste receptor-like chemosensors, such as the laryngeal taste buds, are located. Therefore, we would like to investigate this connectivity. We would like to identify CalB1 expressing vagal sensory neurons forming close contacts with taste receptor-like chemosensors not only in the larynx trachea, but also the tongue and the duodenum. The reason we're very focused on this specific population of neurons is because their function is essentially unknown. And this study would set the foundation for uncovering the role of these CalB1 neurons in the tongue and the digestive tract. Our methods include the use of transgenic mice. We will be crossing 2 mutant mouse lines to then generate a CalB1 Cree TD tomato expressing mouse, which genetically labels CalB1 expressing neurons. We will be euthanizing these mice and collecting tissues including the tongue, larynx, trachea, duodenum. We will then section these tissues and we will be performing immunohistochemistry to detect taste receptor-like cells using specialized antibodies for taste receptor cells. We will then be viewing these sections under a confocal microscope and analyze them through image J and Fiji. Before we began any experiments on our calbindin-1 transgenic mice, we began our experiments on another transgenic mouse line carried in our lab. Here we have an image of the circumvallate papillae taste buds of a FOX2B AI9 mouse, which labels all gustatory neurons. We will be using a FOX2B mouse as a positive control to compare innervation patterns of CalB1 and FOX2B neurons. We also tested common antibodies for taste receptor cells. Here we have 3 different taste receptor markers that we will be using, which are CAR4, TRPM5, and GNAT. CAR4 is a sour cell marker, while TRPM5 is a sweet, bitter, and umami taste receptor marker, and GNAT is a primarily bitter cell marker, which is why co-expression is seen in a subset of TRPM5 expressing cells. Regarding our calbindin-1 mice, we actually have begun a few experiments. Initially, my project was focused on the laryngeal taste buds and the duodenum. However, out of curiosity, we decided to look at the tongue. We found that calbindin-1 is actually expressed in the tongue as well. In the figure on the left, we are able to see 3 strong calbindin-1 signals just from the surface of the tongue. Because of this, we decided to look further into the tongue. In the figure to the right, we also found other strong calbindin signals in the back of the tongue as well. As a result, we decided to shift my project a little to then begin with the tongue, and then we plan to continue down to the larynx, trachea, and eventually the duodenum. As we continue, we're excited to see what other interesting findings we're able to gather. To wrap up, I want to highlight our findings, which is that we found that CalB1 neurons innervate the tongue. We aim to identify innervation patterns in the tongue and the laryngeal taste buds to see if there's any similarities or discrepancies between these taste buds in the tongue and in the larynx. By identifying these innervation patterns, we hope to then uncover the function of these CALB1 neurons in the tongue and in the digestive tract as well, which can be accomplished through live calcium imaging. I would then like to end by acknowledging the STEAM program here at UTSA and the McPherson Lab for their support. Thank you.